It is 6 o'clock, so we will get started. This event is the second event of this week for our Darwin Days celebrations. My name is Rob Ross. I'm Associate Director for Outreach at the Paleontological Research Institution. And Darwin Days is an annual series of events, a week of events that celebrates the field of evolutionary biology. And it's a kind of event that's hosted internationally, but it's in Ithaca. The Paleontological Research Institution has been hosting it together with Cornell University since 2006. And each year we have a different theme. And this year we are absolutely delighted to be partnering with the Cornell University Insect Collection and Department of Entomology to have a series of presentations and events on insect evolution. So this evening, we are privileged to have 4 graduate students in the department who will be talking about themselves and their research and why insects are cool. And but before we get to that, a couple of other things. So first of all, let me briefly cover what's coming up. So I am going to share my screen to show you the Darwin Days events. You have presumably been here to actually register for the event this evening, so you will recognize these. Tomorrow evening is Science in the Virtual Pub with Dr. Jason Dombrowski. And that event will be at 7:30 in the evening. I should also let you know that Science in the Virtual Pub is an event that is held twice a month, always with an interesting speaker in the area of science, wide-ranging series of talks run by my colleague, Don Haas. And so sometimes they're held in a real pub, but during COVID, they have mostly been held virtually. So we hope that you'll join us for that as well. Then on Saturday, for families, there is an event called Insect Encounters at the Museum of the Earth. That is basically going to be activities for families and their children. And then also on Saturday at 2 p.m. at the Theater Cinemapolis will be the film Microcosmos, which is just an incredible visual celebration of insects and the like. Before we begin, I should mention that, so since this is a webinar and we can't just kind of shout out our questions the way we might if this were a Zoom meeting, the way we'll do it is you'll see a little question and answer icon in the lower part of your screen where you can ask questions. And if you have questions along the way that are, so we'll have our 4 speakers will each go in turn. If you have a question for one of them that you think is very specific to that person, then please ask your question then, and we'll take a couple questions after each of their talks. And then we'll take questions after they're all done. And so that could be a specific question, or it could be a general question that you would like each of them to answer. So I will introduce all 4 of them right now, and then and I'll introduce them very briefly because they'll tell you about themselves. So we have with us Megan Barkdull, who is a 3rd-year graduate student interested in complex social traits and genomes and the evolution of ants. You'll hear a lot about ants this evening. Chloe Jelley is a 2nd-year student in the department, and she is also interested in traits in ants and has already done some research on them as an undergraduate. Alan Mata is a 1st-year graduate student, and he works on bee flies in a couple of different parts of the world. And Kyla O'Hearn is a 2nd-year graduate student, and she works on fruit flies in Hawaii and is especially interested in conservation in Hawaii. All right, with that, I'm going to hand it off to Megan.
Great, thank you so much for that introduction. And let me just get my screen up. All right, awesome. So hi, everybody. Thank you so much for being here tonight. Really looking forward to talking to you all. Like Rob said, my name is Megan Barkdull, and I'm a third-year grad student up in both the entomology and ecology and evolutionary biology departments up at Cornell, and really excited to tell you guys about my research tonight. So if you're here watching this panel, then I suspect that you, like me, are probably interested in insect biodiversity. You might be interested in the kinds of insects that you see around us here in the Finger Lakes or wherever you are tonight, or maybe your imagination is captured, by the exotic insects that you can find in the tropics, or maybe all of the above. And I know that I myself have definitely been interested, fascinated by bugs since I was a little kid, catching ladybugs, butterflies in my backyard. I think it's really cool that this week you're screening the film Microcosmos, because I remember watching that when I was probably 4 or 5 with my parents and just being fascinated by the world of insects. So I just really always loved the outdoors, found insects really fascinating even as a little kid. And then, all through high school and into college, I just always really loved biology. So those were my favorite classes. I enjoyed reading about biology, loved biology classes, things like that. But heading into college, right, the only career that I really knew about or had firsthand exposure to that used biology was medicine. And so I entered college actually thinking that I wanted to go to medical school and become a doctor. And it wasn't until a little bit later in my college career as an undergrad that I first got firsthand exposure to the idea that it could be possible to go to grad school and then become a scientist and use biology in that way. So in undergrad, I actually had the opportunity to work in a lab that used various genetic tools to help us understand how we can best protect endangered and threatened butterfly species to protect them from going extinct. So it was really in college that I developed a love of both genetics, which you'll hear about tonight, and then also studying insects from a scientific perspective. And so that kind of ultimately led to my decision, instead of going to medical school, to come here to Cornell to pursue my PhD. And there's a lot of good reasons why we should all care about and be interested in insects, right? For one thing, insects make up 40% of known animal species. So 4 in 10 of all of the described species of animal on Earth are insects. And to put that, another way to put that kind of in perspective in another way, insects are so diverse that if you just picked a random animal species on Earth, you'd have a 1 in 4 chance that species would be a beetle. Literally 1 out of every 4 species on this planet of animal are beetles. So that's just wild. That's huge, huge diversity. And insects aren't just, speciose and numerous. They can really be found playing a role in every ecosystem, from forests, this woolly bear, to agricultural systems, to wasps in this desert, to alpine mountaintops. And there are even some species of insect that spend literally their entire lives out in the open ocean. So they're basically found everywhere on Earth. And so it makes sense then that as humans and as scientists, we would really want to better understand this incredible insect biodiversity that we see all over the planet. And so the way that I kind of think about understanding that biodiversity is through 2 sort of big questions. And the first one is, why do we see particular patterns of biodiversity? And then the second question is, how has evolution given rise to these diverse organisms? So, in other words, what are the factors that explain the patterns we see around us in nature? And then what evolutionary processes in particular have acted to shape insects? Now, those are obviously really big questions. And to explore them across all 900,000 known insect species, that'd be really challenging, maybe impossible. So my research really focuses in on ants as a study system. And ants themselves in particular are fascinating for a ton of different reasons. I talked about the number of insect species. Ants have about 14,000 known species. And I'm showing you, an example of their biodiversity here on the screen in these pictures. They're also really ecologically important. So, we hear a lot about how earthworms are involved in turning over soil and soil processes in ecosystem. Ants actually do that quite a bit as well. Ants can be really important herbivores, especially, in tropical ecosystems. You think about, really iconic leafcutter ants that can just strip trees entirely of their vegetation in a couple of days. So they can be really, really important herbivores in a lot of ecosystems. They can also be important as invasive species and have a variety of effects on ecosystems. So super ecologically important. And then most importantly for my research in particular, ants are social. And so what do I mean when I say that ants are social? Because we probably all, kind of have an idea of what that word means when we're talking about humans, human sociality. But what does that mean when we're talking about insects? Scientists really have kind of a specific idea in mind when they say that insects are social. And there are 3 criteria that we really use to define social insects. The first criteria is that social insects cooperatively care for their offspring. So here in this picture, hopefully you can see my mouse, you can see that these adult ants, the darker colored individuals, are all working together to care for their brood, those lighter colored pupae off to the side. The second criterion is that in social insect species, some individuals are going to reproduce, and then other individuals are not going to reproduce. So in this image, you can see the larger adults, those are the queen ants, and they're responsible for really laying all the eggs for the colony. And then the smaller individuals are going to be workers that don't reproduce. And so we call this reproductive division of labor, and it's really a fundamental characteristic of social insects. And then finally, in social insects, different generations are going to be overlapping in time. So, you can think about a multi-generational household for us humans, parents, children living together. Just like that in social insects, the queen ants that you see here are the parents of the workers and pupae, and you can see that they're coexisting in time. And then one more thing I want to mention that isn't so much a definition, a defining component of sociality, but it is a really important and interesting aspect of sociality in ants, is that ants can have multiple types of workers or multiple castes. So in other words, not only are female ants divided into reproductive queens and non-reproductive workers, in many species, the workers can then be further subdivided into categories like workers and soldiers, or we also might call those minors and majors. And so because of this sociality, ants have really been quite heavily studied to try and answer the question, what genetic changes cause insects to be social? So what's causing this sociality? And in other words, how does evolution take us from solitary wasp-like insects like the one I'm showing here to insects like ants that can have really complex social structures? Some ants, their colonies have millions of individuals in them. They could have hundreds of queen ants. Ant colonies can span really large geographic ranges of like hundreds of miles. So how do we go from this solitary insect to these really complex social structures? And a lot of genomic research has been done on this topic. And that's allowed researchers to identify some possible mechanisms underlying this sociality. So that could be things like there's some evidence that there could be changes in the timing of particular genes being expressed. There could, in some cases, be the emergence of entirely new genes in these insects' genomes that help them be social. But I'm actually interested for my own research in kind of a slightly different question. So instead of asking how genes can cause changes in sociality, I really focus in on understanding how sociality causes changes in organisms' genes, so kind of the flip side. How does being social cause changes in genes and in how genes evolve? And so while this first question is pretty well studied, the second question, my question, has received comparatively less attention. I think it might seem a little bit less obviously flashy, less obviously interesting, but it really is an equally important piece of the puzzle when it comes to understanding sociality and evolution, right? It's really important to have a complete picture of what's going on both sides of the story. And so to do this in my research, I basically compare organisms' genomes to one another to try and associate different social traits with particular evolutionary patterns. And so the kinds of social traits I'm thinking about here are going to be things like how many queen ants are there in a single colony? Do the workers ever lay any eggs? Are they totally sterile, or are they maybe capable of a very limited degree of reproduction? Or are there multiple types of worker ants in the colony? Like I discussed, do we see that kind of subdivision into majors and minors? And so I want to understand, how do these various social traits impact evolution in ants? And so to answer those questions, I am currently using the genomes of 23 different ant species that have been sequenced. They're out there, they're publicly available. Any one of you could download them and look at them. It's pretty cool, the data sharing that goes on. And I take those genomes and I basically compare them to one another. And then I use a bunch of different statistical tools that let me measure and then compare the patterns of natural selection across my different species. And then I can associate those patterns with the different traits that I'm interested in. So just to give you just a touch more detail, a little bit more of an understanding of what that looks like, you probably all know that gene sequences, right, are going to be made up of 4 different nucleotides, A, T, C, and G. So that's what I have shown on the screen here. And then the tools that I use basically line up the gene sequences from each of my species, and then they compare them so that, you know, you can see how they're different, how they differ from one another. And then depending on the patterns of differences that I see or that these programs detect, we might be able to infer that some species, you know, have a lot more differences compared to the others. And then we could perhaps conclude that they've experienced a different amount or a different kind of natural selection. And then we can correlate that pattern that we see, again, with those different traits that I talked about. And so what have I found so far in my research? I've found that species tend to experience more positive selection when their colonies have just a single queen ant, as opposed to, you know, species with multiple queens in the colony. When worker ants are capable of some reproduction, species seem to experience much less positive selection than do species with totally sterile workers. And then finally, that third trait, species that have multiple distinct kinds of worker ants seem to experience less positive selection than species with just a single kind of worker ant. And so overall, the big picture takeaway that I'd like you guys to have is that, yes, it does appear that social traits do impact evolution. And it appears that they impact evolution in kind of a variety of different ways, right? So it's not one size fits all. It depends on the trait that you're looking at, how it's going to influence evolution. And going forward, this research may help us better understand how evolution works, you know, in solitary versus social species, like I've been talking about, or even how evolution might be impacted by transitions like the one between unicellular organisms and multicellular organisms. So you can kind of draw this analogy between solitary insects and social insects, and then unicellular organisms and multicellular organisms. And ultimately, you know, using organisms' genomes in this way, these comparative genomic methods, can be a really powerful way of better understanding all the diversity of life on our planet around us. And so with that, I want to say thank you guys, and I'd be happy to take any questions.
Okay, do we have any questions already?
There's now a question in the chat of what's an example of positive selection in ants.
Okay, yeah, that's a great question. So positive selection basically means that evolution has favored a new form of a gene as opposed to the pre-existing form of that gene. And so I've found a lot of evidence for natural selection really across a broad variety of different kinds of genes. I could get into the really technical details of like what those biological categories are, but I'm still sort of working on the piece of the puzzle of what do those particular biological categories mean for what's going on in evolution in ants. So I don't want to make too many claims about that right now. But it basically means that natural selection is selecting for a new form of a gene as opposed to the previous form of that gene.
Okay, and we have a couple more questions. What do you think is the social trait that will be most important for shaping their genomes?
Oh, that's a great question. I actually think one trait that I've thought about that I'm not actually analyzing, but other researchers are working on, is the size of the colony. So that can potentially play a pretty important role in how their genomes evolve. I also think that having one versus multiple casts that I talk about, that's a pretty fundamental difference between different species of ants, and that's probably going to be playing a pretty important role in how they evolve. Great question.
Okay, and where can we find your research?
Yeah, so none of this is published yet. This is all still kind of a work in process. Those tests that I talked about take a really, really long time on a lot of computers to run. So keep your eyes peeled. And you can also follow me on Twitter if you want to kind of see a little more behind the scenes as things come along. Okay,
so we'll take 3 more questions here, good questions, and then we'll move on to our next speaker, and then we can hold some for the end. Merv asks, does natural selection act on genotype, or can natural selection act on the genotype?
So the way that I would describe it is that natural selection is going to act on the phenotype. So how the genes are actually expressed into an organism's, morphology or behavior or whatever. Natural selection acts on the phenotype, but that phenotype is ultimately a product of a combination of the environment and the genotype. So ultimately it does kind of have that feedback effect onto the genotype, but it's not quite that it's acting directly onto the genotype, if that makes sense.
And you may have said this, but could you just give a 1-sentence description of the difference, what a genotype and a phenotype are?
Yeah, absolutely. So a phenotype is everything, physically about you. So your morphology, your behavior, all of those kinds of things. And then the genotype is the gene sequences or the genetic code that is within you that gives rise to those traits.
Okay, thank you.
Yeah.
How many generations do you have to study to see a change?
Oh, that's a great question. So I'm really just looking at a snapshot in time. So I have one genome from each of these species. And so I'm not really looking directly at how things are changing over time. And if you were to do that, so if you were to, sequence a bunch of genotypes from the same species over time, it would really kind of depend on the strength of natural selection. So if natural selection is operating really quickly, really strongly, that change could take place in just a couple of generations. If natural selection is a lot weaker, it could take hundreds, thousands of generations.
Okay, and one more question before we go to our next speaker. Great talk. Of the 25 or so species genomes that have been sequenced, are they all from different habitat types? Does this seem to influence sociality? Do you see a pattern?
That is a great question. I actually literally right before this talk I was on Twitter and I saw a cool paper that I'm excited to read that is correlating some of these traits that I'm looking at with different environments, and I think finding an impact of like desert type environments. I'm not directly looking at habitat in my particular study, but these species that I'm working with do come from a variety of different habitats. Yeah, great question.
Awesome. Okay, thank you so much, Megan. So we'll, and if you have more questions for Megan, we'll see her at the end. Our next speaker will be Chloe Jelley.
I'll just share my screen. All right. Hi, everyone. I'm Chloe Jelley. I'm a second-year PhD student in the Moreau Lab as a part of the entomology department at Cornell. And I'm excited to talk to you guys today about ant evolution and diversity. So my focus is almost entirely on ants. I find them extremely interesting, and there's so many things about ants that are fun to investigate and examine. So today I'm going to be talking about 3 major things. So first, I'm going to start with my research I did in undergrad, looking at the potential drivers of ant eye diversity. And then I'm going to talk a little bit about my road to grad school, and then the kind of questions that I'm currently interested in. So this project that was looking into the drivers of ant eye diversity was kind of inspired by being in the lab, actually, and looking at specimens in ethanol. And what I was doing was what happens when entomologists go out in the field. A lot of the times you're collecting a large mass of insects, and then either with ants, we drop them in ethanol to preserve them, and then we bring them back to the lab where we dry them out and point mount them onto a pin, and that way we can preserve them and look at them for a really long time. So the way I was kind of introduced ants wasn't really going out into the environment and looking for them. It was actually interacting with preserved specimens and databasing them and cataloging them. So basically I was looking at this vast array of ants and not really seeing them in their natural habitats. And it made me very curious about why some ants had just such different body types and shapes and forms. And I became very, very interested in the vastly different eye shapes and sizes and heights and the fact that some ants are entirely eyeless. So I began doing a lot of research looking at all these ants that I was seeing in the lab and looking them up and seeing the kind of literature that was around them from biologists that have been in the field and had observed them. So I came up with a few questions, and I also came up with a few potential explanations for that diversity. So my two questions were, is there a trade-off between the antenna and the eye? And this comes from the idea that sensory organs like the eye and the antenna, which the eye is obviously the visual organ, whereas the antenna is more of like a chemical tactile. So they're using it to touch their environment, to take in like the chemicals around them. So the idea that these sensory organs could be potentially costly during the development of the immatures, like the idea that one could be costly. So the way that you look at this is if there is an increase in eye size or eye height or eye area, is there a decrease in the length of the antenna and vice versa? So let's say an ant is living in a subterranean environment, so under the ground, you wouldn't necessarily need a large eye to be able to navigate that environment. So ants like that may have a reduced eye or no eye at all, and they may or may not have a larger antennae. So that was one of my possible explanations for why there would be such diversity within the eye shape, size, area. And then the other thing I was thinking about is eye size just directly related to the ecology? Is it directly related to how these ants are navigating their environment? Ants can occupy many different types of environments. Like I said, they can be subterranean, but they can also live in the tree canopy. So there's ants that almost live their entire life foraging and navigating the tree canopy, nesting in twigs. So there's these vastly different lifestyles that ants can follow. So it would make sense that the way that ants live their lives would strongly influence what visual or chemical or tactile systems that they have. So my approach was to take a whole bunch of measurements of the eye and antennae. I actually came up with a few very, very specific measurements in order to quantify how much that the ant is investing in the eye versus the antennae. And then I also kind of mentioned this before, I scanned the literature looking for all the information that I could get on their ecology, how they are interacting with their environment. And for this, I focused on two major things. I focused on the diet. So ants can have a very vastly different diet. So some are strict predators, whereas others are herbivores. Everyone knows the leaf-cutting ants who are fungivores, and they eat entirely fungus. And then also the habitat, I looked into the habitat. So whether or not they are foraging arboreally, under the ground, or anywhere in between. So to kind of drive home how insect vision works, because I think there's a lot of misrepresentation of insect vision in movies, the classic, if they show you a movie and you're seeing it from a fly's perspective, you see a bunch of tiny little images. But actually it's not quite like that. And insect eyes are made up of these facets that we call ommatidia, and the ommatidia combine to create the resolution of a picture. So you can see on the right of my screen, and this is just kind of like a oversimplification, but I think it gets the point across. So you can see this 70 pixel resolution is very, very pixelated, and you can liken that to an ant that has 70 facets. Or if you increase to 800 pixels, that could be 800 facets. And then I also put in the human resolution to kind of compare. So after I took all these measurements of the eyes and the antenna, the first step was to map it onto phylogeny. So a phylogeny is kind of like a visual representation of the evolutionary relation of what we think the evolutionary relationships are of ants or any organisms, but in this case, it's ants. So I've mapped the ommatidia density, which is basically the number of facets per millimeter of eye. And then I also, on this other side, I have mapped the eye area. And this is really just to show the diversity that there is across these ant groups. And these are just the taxa that I measured. So this is a very, as Megan mentioned before, there's over 14,000 species of ants, and I could only include as many that I had access to in the lab. I also mapped the trophic level, so whether or not it was a predator and omnivore. And then I also have the foraging niche. So this is the habitat in which they're navigating. So we have the leaf litter, arboreal, hypogeic, which is below ground, and epigeic, which is above ground. And here I just wanted to show again just the diversity of the different placements of eyes and how it might be related to ecology, and just to show, I really want to drive home the diversity of how different ants are, because it's definitely not something that I knew before researching them. So I just want to share that with you guys. So the results of my study, focusing on the first question, is there a trade-off between eye and antenna investment? And what I found is maybe. There's some relationships that I found between the scape length, which is the first segment of the antenna, and various eye measurements. But there wasn't relationships with all of my measurements. And I think with these kinds of studies, obviously, the more different species that you can include, the better your results will be. So I just had a small subset. So you never know if increasing the number of species in your study would make the relationships that you see stronger, or if it would make the relationships that you're seeing completely disappear. And then the answer to my second question is eye diversity related to the ecology? Not really, but we can't say no. There's a slight trend for there's more eye investment in omnivorous ants. So these are ants that are both they're both eating other arthropods, other insects, but also consuming more plant-based foods. So that's my research from undergrad. It's published, so I can, if anyone's interested in learning more about that or asking more questions about that, I can put the name of the paper in the chat, but I'm going to move on to my road to graduate school. So you've heard me talk a lot about how I'm interested in ants, how I think they're super cool, but I was definitely someone who had to be sold on how interesting insects are. I started in the lab because I thought I was a general biology student at the New Jersey Institute of Technology. And I thought I wasn't really sure what I wanted to do. So I thought the next step would just be to volunteer in a lab and see if I liked it. And I really had a lot of fun looking at all the different types of ants, and just it amazed me how they really come to life under the microscope. I would have never pictured that the things in my kitchen growing up would be so interesting to me. So as I got more and more interested, I decided to look for more opportunities to see if I really enjoyed it. So I got the opportunity to go to Madagascar and do field work. The field work that I got to participate in was arboreally sampling in the dry forest in Madagascar. So we used rope climbing to go up into the trees and to assess the ant diversity there. So that was, I think, a really another super important step on my way to being like exposed to science, but also to realize that science is something that I could do. And yeah, I also just had a lot of fun after only seeing the ants in the lab previously, to see them walking around the trees. And I was able to recognize species that I had only previously seen in the lab. So that was really cool. And so now I am a graduate student at Cornell, and it's been a super interesting journey, mostly dominated by COVID, but I'm certainly learning a lot, and I have a great lab to interact with. And that leads me to the questions I'm currently interested in. So I know Megan talked a lot about how ants are eusocial, and how this is one of the reasons that a lot of people study ants and a lot of people are interested in ants. And one important aspect of that is that you have these single reproductive units. Like each colony has a queen that is the reproductive, and then the workers are sterile. But in order for this kind of system to work, you need a system of defending yourself too. So the workers, one aspect of ant biology that I've really been focusing on is aggression and various forms of aggression. So you have like this very vastly different responses to nest disturbance. Everyone who's here in Ithaca may not really think of ants as being a super aggressive insect. I don't think anyone would be super scared of a tiny little ant colony in their backyard, but I know people, if anyone here is from the South or has encountered fire ants, everyone knows you should stay clear of those. So there's this really interesting variation in aggressive behavior in ants. And I'm really interested in kind of breaking that down piece by piece and looking at if there's any traits that could be correlated with aggression, with this variation in aggression, and then try and break it apart and really try to understand where this variation is coming from. And that's all I have prepared. Thank you all for being here and listening to me speak. And I think I'll take a few questions.
Awesome. Thank you so much. Yeah, we have a couple of questions already. And the first one is from An. Are ants that glide driven to evolve better visual systems over other senses?
That's a really good question. I know some ants that glide would be Cephalotes, the turtle ants. And from how I'm picturing them, they do have pretty large eyes compared to their body size. I haven't looked into, I don't know if there's enough ants that glide that you could do a comparative study with that, but a lot of arboreal species have really high investment in eyes. So, and then you wouldn't necessarily need to glide if you weren't living up in the tree. So I think that would probably be a fair assumption to make.
Cool. Okay. So next question. So many placements of eyes and sizes in ants, exclamation point. If you had to guess what might explain all this variation that you did not measure, what do you think might be important?
One thing I wish I had included in my study is the time at which ants are active. There's a lot of ants that are active during the day or active during the night. And I think this may have a really large impact on eye investment.
Okay. Two more questions. Was there any noticed correlation between eye presence and social weight of face construct? You might have to explain to us what that means. Like how paper wasps recognize other wasps via color patterns on the face.
I know ants use chemicals more to recognize individuals, and it's not necessarily individuals that are super important to ants. They recognize, basically they have the system of recognizing whether they have a colony mate or not. And it's basically, if you're not in your colony, you're potentially a danger to the colony. So what they have is these chemicals on their cuticle that they use to kind of check if you're okay, kind of like an ID card. And that is all, that's all chemical. I'm not sure if we know if any ants use visual systems to recognize other ants. There are some ants that have really high investment in eyes that are known to like watch people as they go by, but that's as much as I know.
Okay. And finally, ant aggression. Does that mean you'll be hosting ant fight club experiments for your research?
Most likely, yes. I'm currently in the stage of my research where I've just been reading hundreds of papers that just describe ant fights. So if you ever want to get into that literature, it's quite entertaining at times to read the way that ants are described.
Okay, very cool. All right, thank you so much, Chloe.
Thank you.
All right. So Alan, you are next.
Hi, everyone. Today, this evening, I will be talking to you about how my path was from as a child to now in a PhD program, and then talking a little bit about my research that I did as a master's student, and then what I'm currently going to be doing as a PhD student. So I was born and raised in California, in the California coast, and I was always really intrigued by the biodiversity around me. And so basically, I just was really fascinated with biodiversity. And I remember when I was like, I think this was like in first or second grade, I brought a insect identification guide home, and my mom was just worried. She was like, "What is this?" And lo and behold, I ended up becoming an entomologist. But my actual fascination in science didn't really start till I was in high school, and essentially I had a really good science teacher, and she pushed me to question everything and to observe the natural world. And so I ultimately, at the end of my senior year, I decided to go to Chico State University in California. And I came in, ironically, as a nursing major. And within, I think, 2 semesters at that point, I was like, this is not for me. So I ended up taking an ecology and evolution course, an introductory course. And after I finished, I immediately changed my major to biology. And at that point, I knew of an entomology course that was being offered through the department, but I wasn't at the right grade level at that point. So my junior year of undergrad, I took my first entomology course, and I was the only one in the course that was collecting a lot of flies, and everyone else was collecting beetles, butterflies, and other insects. So my fascination for flies at that point led me to pursue a master's degree through San Francisco State University and the California Academy of Sciences. So there I focused on the research of bombylians, which are commonly known as bee flies. There are roughly 5,000 species described worldwide, and they have a very Afrotropical region distribution. They are commonly known as bee flies because they do, many of them mimic bees and wasps. And the reason for this is because they are parasitoids. So they will parasitize a host and they will ultimately kill it. They are also known to be hyperparasitoids, which is they parasitize other parasites, which is pretty interesting. And they will parasitize as well solitary bees and wasps. Solitary bees, I think that's what they're highly known for. And so for my master's project, I did two things. And the first thing was to curate the Madagascar bee fly collection that was present at the California Academy of Sciences. And my goal there was to just eventually describe new bee fly species. Now, unfortunately, this was happening during as the pandemic started, so that project did not unfortunately fold out the way that I wanted it to. So I'm picking that up for as a PhD project. But one other thing that I did is I described 7 new species of Exoprosopa from Madagascar. And the way that this came to be was essentially Erica McAllister from the Natural History Museum in London. She messaged me and she was like, well, I have all these boxes of undescribed Exoprosopa species. Would you like to take a look at them? And I could not say no. So I described 7 of them, and I'll show you some of those at the end of my talk. And now as a PhD student, I am going to be looking, I'm still going to be working on bee flies, but I'm going to be working on a genus called Poeciloanthrax, and I'm going to be looking at a couple of things. So I want to do a revision of the genus, and this will be by inferring a phylogeny. Not only that, phylogeny and morphology, and I also want to look at the distribution of members of the Poeciloanthrax in the southwest and of the desert of California. Lastly, as I said, I will also be doing a project, hopefully bringing back what I started as a master student and finishing that up as a PhD student. And I think that's basically it for what I had to talk about. I wanted to show you guys a few images of the bee flies that I actually described. I'm going to show you guys 3. Give me 1 second. So this one's actually named after Charles Darwin, Charles Darwin. And as you can see, this one's quite beautiful. And these are all the ones that I'm going to show you are from Madagascar. So here we have the dorsal view, and then we have the lateral and the face view. And you can kind of see it's kind of waving at you with its tarsi, which is quite adorable. Oops, sorry. The next one is Exoprosopa floresi. And this is actually named after my mom. This one's actually quite a really beautiful fly. As you can see, it has some orange on the sides of the abdomen, really beautiful coloration on the wing. And again, we have the dorsal, lateral, and face view. And then this is named after Erica McAllister. So this is one of my favorites, actually, Exoprosopa McAllisteri. We have the dorsal view here, and we can see that it actually has a proboscis, which is double the size of the, almost the size of the head, actually. We have the lateral view here, and then we have the face view. And what I love about this fly is this, in the fronds, in the fronds, there are these beautiful gray scales, as we can see here. Now, the stuff on the sides of the eye, those are just materials that unfortunately were stuck to the fly when they were collected. I couldn't take that off because these were specimens from like the 1950s, and I was afraid of actually breaking a head while doing that. So I just let that be. And I will say that in Madagascar currently there are 18 species, and all these ones that I just showed you are not published yet. But once this work gets published, there's going to be an additional 7 more species of bee fly in Madagascar. And yeah, I want to thank all of you for joining me tonight, and I will take any questions.
Very cool, thank you.
Mm-hmm.
So we have a couple questions already, and I wondered if I might ask one first, because I think, for those of us who have never described an insect species, I wondered if you might just take a moment to say, like, what does that mean? It obviously means more than writing it down in your notebook. Like, you are making a record sort of for all future scientists about what these species are. Could you describe that a little bit?
Yeah, so when you describe a species, you're basically describing the entire anatomy of the insect that you're looking at. And not only that, you're also making what we call a dichotomous key, which is a way that scientists can then go and use a key to actually identify the insect that you're looking at. It takes some time because you also, not only do you have to photograph, but you have to sit down and look at not one specimen, but multiple specimens to do this. And for me, it was a 2-year project. So
Yeah, cool that you get to do that. And so both questions are sort of along these same lines. The first question that's in the Q&A says, so cool that you have described new species. Do you think you will be able to describe more during your time at Cornell? So so jealous.
Absolutely. I really hope so. I mean, there's still a lot of diversity out there that's not described, especially in the desert, in Arizona, closer to the Mexican border. There's definitely a lot of more diversity down there to describe. And I might find some more new species, even in the genus that I'm going to hopefully revise, because I'm planning on, again, inferring phylogeny. And it might be that some species might be cryptic species, and we might have a new species that we didn't know about before.
Okay. And follow-on question, I always wondered how scientists come up with scientific names. How did you come up with the names? Did your mom cry from happiness or fear since you shared she didn't like insects when you were growing up?
So the way that there is a code of how to name species, but the way that I did it was I inferred with my former PI on what she thought was appropriate, and I made sure that for the species that were already described, that the species name was not taken. From there, that was what I chose. Now with my mom, she was quite delighted when she saw how beautiful it was. She was, again, when I was younger, she was a little bit horrified that I brought that book, but hey, it all worked out in the end, right?
All right, excellent. Any other questions for Alan? If not, Kyla, you are up.
Yay. So thanks everyone for, I guess, sticking around this long. Again, I'm here to finish us off with this whole panel, which, thank you for allowing me to be a part of. So I'm just gonna, hello. So I'm just gonna be talking about my research a little bit and I guess how I really got here, because the trajectory was really weird. So hi everyone, my name is Kyla O'Hearn and I am a 3rd-year PhD candidate in the O'Grady Lab in the Department of Entomology at Cornell University. And I'm also a 1st-generation student, meaning that neither of my parents got their bachelor's. And I mention that only because this means that when I was accepted to my undergraduate institution, the University of Kentucky, I had absolutely no idea what I was doing. I remember not even having a laptop, and I remember going to the library every night just to do my homework. And I think it was the lack of this exposure that really shaped my trajectory into academia. So actually, despite my title, Kyla O'Hearn, PhD candidate, I actually started off, I think you saw before, as an animal science major. There I am, so happy and so full of life holding that little lamb. But I actually started as an animal science major because, I love animals and I honestly didn't know what else was out there. But to be quite frank with you, I was underprepared and overwhelmed. My GPA after my first semester was, I had to look it up, a 2.091. It wasn't the best situation to be in. I was on academic probation. I was really scared that I would not finish college, but that all changed in a positive note when I took my first general entomology course, very similar to Alan, who was also in my lab. And I really found my passion there. And from there, I was exposed to laboratory research and I began working in all these different labs. I started off in Dr. Jen White's lab looking at aphid endosymbionts, and then moved on to looking at landscape and how we can really use different plants and different orientations to attract monarch butterflies. in Dr. Dan Potter's lab, and then I went into Dr. Dave Gonthier's lab looking at sustainability and agriculture systems. So I was all over the map, basically. And I guess after getting this exposure, well, I never really looked back. I ended up changing my major, and I think I changed my major in the fall of 2017, graduated in fall of 2018, and started at Cornell in the fall of 2019. So this was all definitely a whirlwind experience, but I like to think that everything happens for a reason. And, you know, I'm here. So when I came to Cornell, I knew from my prior experience in different labs that I was interested in general insect identification. I was interested in taxonomy, which is the classification and naming of organisms. And I was interested in systematics, which looks at the evolutionary relationships of organisms in between different organisms. And luckily for me, in Dr. O'Grady's lab, I am able to incorporate all those interests with a really, I'm very biased, but a really cool system, which is the Hawaiian Drosophila. And I'm sure many of you are somewhat familiar with fruit flies. You might see them all over your house, maybe near some overripe bananas. But the Hawaiian Drosophila have a really cool origin story, if you will. So basically, a single fly was likely blown over to the islands, likely due to a storm, and this happened approximately 25 million years ago. And just from that one colonizational event, those flies were able to quickly speciate due to the variety of plants that you'll find all throughout the Hawaiian Islands. And today, and I should mention, probably one of the coolest things about this colonization to the different islands is they colonized by this sequential formation of the islands. So starting at the oldest island and then kind of moving to the newer islands in a sequential fashion, which provides this really cool geological timescale to study these flies. And actually today you can find about 1/3 of all species of Drosophila solely within the Hawaiian Islands, which is absolutely incredible. And so with this system, I look at their ecology, their evolutionary history by looking at, phylogenetic trees. I also do species descriptions, as Alan talked to you about in his talk. And then I also study them, and this is probably the more closer to my heart, I study them from a conservation perspective, which is what I'm going to be focusing on in the second half of this. So I mentioned that these flies were able to speciate very quickly throughout the islands, and that is largely due to the wide variety of host plants that are available on the islands. And many of these host plants are similar to the flies, exclusively found on the Hawaiian Islands. The problem with that is these plants themselves can be very vulnerable to habitat modification due to the large prevalence of invasive species. Because the Hawaiian Islands are such an isolated system, they can be more prone to really getting hit from all different sides from invasive species. And so there are weeds that will outcompete these plants, and there's feral ungulates such as wild pigs that will actually trample the plants. And once trampling the plants, it'll make room for these weeds to grow. So it's just, like I said, hit from all angles. And these flies are so tightly linked with their host plant that when the plants go, the flies will go as well. So as you can imagine, it's a very fragile system. And I plan on further studying these impacts by looking at historical data that was collected in the late 1960s. Basically, a man named J.P. Murphy took surveys of Drosophila every 2 weeks for 2 years. So lots of data in the Waikamoe Forest Preserve. And with this data, I'd like to not only analyze the data and look at, trends of diversity and abundance, but also conduct a series of surveys using his methodology to see if I see any changes in that abundance and diversity, but over time. And I'll also look at, temperature fluctuation and humidity, precipitation to see maybe that could cause any impact in that as well. And, it's hard to say for sure what I'll see as I haven't started analyzing the data yet, but, I have a good idea, but I won't spoil it yet. And as I mentioned earlier, habitat modification can drastically affect the prevalence of Hawaiian Drosophila. And I was actually informed that since that historical survey was done, a weed called invasive ginger, which is actually that second picture in the middle there, has absolutely dominated that landscape where the surveys were taken. So in all likelihood, I imagine the number of flies that I will find when I eventually go back to the Hawaiian Islands, post-COVID maybe, will be very low. But I'm having some hope that, maybe there'll still be some out there. And yeah, this is just a shot of what that data looks like. He was very organized, luckily for me, and his handwriting is really easy to interpret. So basically, I'll just have to maybe update some of the species names, as I'm sure a lot of them have changed. And like I said, analyze that data and just to get an idea of what's where and what I can expect to see. And I guess to finish off, I really love what I do. And I hope to inspire the next generation of scientists, as well as those who just don't believe that they're good enough to be in academia. I like to share my story of how I got to this point, because I don't think it's, a very traditional trajectory of how people get into graduate school. But nonetheless, it is my story, and I hope it does inspire, the next generation of scientists. And I'd be happy to take any questions if anyone has any.
Very cool. We have several questions already, actually. And the first question, Kyla, if only one individual fruit fly was blown to the Hawaiian Islands, how is their reproduction?
That's a fair question. I should mention this was a female that was impregnated, that was blown over to the islands. And it's very rarely is this kind of colonization event actually successful. So, it's very hard to believe a very itty bitty teeny tiny fly was able to make that travel, that journey all the way over there. But it was just a perfect series of events, and it happened, and it's a really cool study.
Okay, what do you think is the biggest threat to Hawaiian insects? What can we do to help?
Biggest threat is likely those feral ungulates that I mentioned. So a while back, when they began incorporating farming and grazing on the Hawaiian Islands, that's how these animals were released, unfortunately. And so now they roam free. Like I said, they trample those native plants that are essential for the flies to reproduce and feed their larvae. So there are conservation efforts being made, not just for the flies, but several other, fragile species, including putting up fencing in some of the more preserved areas to keep out the ungulates. And there has been an effort to reforest some of the areas, replant, although it's not completely well known as to whether those efforts are working or not, but there are efforts being made.
Yeah, super challenging. Okay, next question. I didn't know that 1/3 of all Drosophila species are found only in Hawaii. Have you been able to go collect them in Hawaii? If so, what was it like? Hashtag collecting bugs in Hawaii is so cool.
I definitely can't complain. Yeah, we made a trip. Oh, geez. When did I start? So I think it was January 2020, right before the pandemic hit. It was my first time in Hawaii, and it was an amazing experience. it was interesting because actually the entire trip, it rained the whole time. And if you can imagine people swinging butterfly nets in the rain, you're not going to catch very much stuff. But I haven't been back since then because of COVID, but I'm hoping to go again soon-ish, and hopefully we'll have a better data collection then.
Is there a particular island that's most interesting, or do you visit all of them?
We visited most of them. The island of Maui is particularly interesting to me. Just fewer people, cooler bugs.
Excellent. What is your favorite Hawaiian Drosophilid?
Oh, my advisor is not going to like me for saying this. I think the picture wings are really cool. They're these big, you all should look it up, these big ornate looking flies, as their common name indicates. Their wings are absolutely stunning to look at. And I think because they are these big ornate flies, if you look at, from a conservation perspective, the threatened or close to extinct insects of the Hawaiian Drosophila are all picture wings. There's 11 of them that right now are considered threatened or endangered. And I think the smaller ones aren't as well noticed, but regardless, they're really cool flies to look at.
Okay, and there's a comment following all of that that says, "Thank you for sharing your story." Hashtag inspiration.
Yeah. Thank you for having me. This is great.
So I want to open the floor now for general questions to anybody. And I know that we've been here for a while and we've had a lot of really great questions and terrific answers as well. But if I would actually like to start this, and I want to ask, so to each of you, that you have shared experiences with going into the field, and then evidently each of you are also working in the lab. And I wondered if each of you could just take a moment to say, which of those two do you like better, or do you like them the same but differently? And Kyla, why don't we just start with you?
Oh yeah, that's an easy one. I definitely prefer being in the field. It's Hawaii.
Right, yeah, you have the right field area. Alan?
I love being out in the field. I mean, you cannot go wrong being out in the desert. You get all this beautiful diversity.
And what a contrast.
Right.
And tropical forest in Hawaii. How about Chloe?
I feel like it's a really hard question for me because a lot of my research is inspired by insect collections, and I find it just so fun to open a drawer and just see what's inside walking through. But there's something really special also about being in the field and seeing the organisms that you're interested in interacting with the environment. So I'm going to cheat and not pick one and say they're both great.
Okay. And Megan, are you going to buck the trend, or what do you think?
No, I think I would really have to agree with Chloe. I agree that, being out in nature, that's originally what got me interested in all of this. And there's nothing like that. But I do also really enjoy lab work. And I'm also going to put in a plug for computational research, which is what I've been doing for the past years. I don't always love sitting in front of my computer screen, but you can also, just answer such cool questions that way. So I would be torn.
Yeah, that all of these fields require some kind of computer analysis. Okay. Does anybody else have a general question for everybody, or did you have your questions answered already?
Probably one more in the.
Okay. But did I miss it?
Yeah, I think it says, does the world need more entomologists, or are you and your colleagues at Cornell covering all the work that needs to be done?
Yeah, I didn't scroll down and see that one.
Yeah.
Let's see,
anybody want to volunteer to just take that?
I guess I can say that we need more entomologists. We only specialize in certain genera, species, right? And some groups are very large, and you just cannot specialize in all those groups within whatever you're trying to study. So the more entomologists, the merrier.
Yeah, I think the insect species to entomologist ratio is pretty high.
And I would also just add on to that and say, not only are insects super, super diverse, and we need more people studying that diversity, our field has also, really historically maybe lacked some diversity. And so we really need more folks from diverse backgrounds joining in and looking at insects with us, for sure.
There are actually a couple more questions here that I didn't notice that I guess could apply to any of you. What advice would you have for collecting nocturnal species?
Ooh, I'll take this one. Nocturnal species, a lot of us for fun, we'll go out at night at a really good spot. Going by water can be really helpful, and putting up a black light, so like a UV black light, and a sheet, a white sheet, and hanging it up and just waiting, and a bunch of stuff will come to it. It's a lot of fun. Highly recommend it.
And we'll hear about that tomorrow night. Yeah. Any other thoughts on that? Okay. If you had, so this is applicable to everybody. If you had to pick a favorite insect that was not a fly or an ant, what would it be?
So my undergraduate research, like I talked about, was in butterflies, and butterflies still have a really special place in my heart. So I would probably pick one of the two species that I worked on as an undergrad. It's called the Dakota skipper. They are super endangered. They live in tall grass prairies in the Midwest, which are a really, really threatened ecosystem, but they're just the cutest little butterflies. They really look like little teddy bears with wings. I highly encourage you to look them up. So I think that would have to be my pick.
Anybody else want to take that one?
I think one of my favorites is the pipevine swallowtail. It's a beautiful butterfly that's found in California.
2 votes for butterflies. Others?
That's fine. I'll be the weird one. I think bed bugs are really cool. I don't know, this insect that has co-evolved with humans, and it has all these adaptations where we don't know they're there. Like 1 out of 3 people don't even have reactions to them. I won't go too in, but I had one feed on me just to see what it was like. It's not any different than a mosquito bite. It's insane.
You're an amazing person. Okay, who did we miss?
I think the insect that comes to mind for me is dragonflies. I just think they're so cool how their immatures live entirely in the water, and I just think they're cool.
Okay, well, we'll make this next one. This will be the last and appropriate question of the evening. You've all done just a wonderful job this evening explaining what you do and talking about your research and how you got here. What entomological outreach things would you like to promote? I'm going to start with one, even though I'm not one of the panelists, and that's the fact that the Cornell University insect collection is going to be featured in a wonderful new museum exhibit at the Museum of the Earth, opening on March 28th. And so I hope that people in the audience will come to that, and that a lot of the fine work that's being done at Cornell will be featured there. And with that, I will hand it to our actual panelists.
And I should put in a plug that Megan, Chloe, and I are all participating in the making of that. So definitely check it out. I should also mention that Chloe and I are actually the co-chairs of the outreach committee in our graduate student association in the department. So our big event that we haven't had in 2 years is Insectapalooza. Awesome event. We get, hundreds of people coming, and we get to show off all of our research. And I'm hoping that, once COVID is over, we get to put that on again, cross our fingers for maybe this fall, but we'll see. But definitely stay posted about that.
It is such a great event for anybody who has not been. Insectapalooza, you got to go. It's got to be on your bucket list for being at Cornell. Okay, there's a note in the chat about what's coming up with respect to Darwin Days and some other things. And I want to thank our panelists, especially, who are wonderful. But also thank you for all the attendees who came on a winter evening, even if virtually, to do this with us. So thanks, everybody. Mhm.
Thanks, everyone.
Thank you, everyone.
Thank you all. And we'll see some of you tomorrow night.